Hello students, this is a recording for Biology 1406, Chapter 5 in the OpenStax Biology textbooks book, Structure and Function of Plasma Membranes. Alright, so the plasma membrane, as you'll recall from the previous video and lesson, is also synonymous with cell membrane, same thing. Plasma membrane, cell membrane, same thing. And it defines the perimeter of all living cells. It also, the plasma membrane term, includes membranes of the intracellular organelle system. And so that manages, the function of a membrane is to control outside versus inside, the contents of the inner compartment versus the world outside of that compartment, and also to regulate the material that moves in and out across that membrane. A lot of that is done via receptor proteins or some mechanism that is able to interpret extracellular conditions and initiate intracellular responses. So you'll recall that the plasma or cell membrane, but membranes, are a phospholipid bilayer, which is a much more detailed term. And here is the phospholipid bilayer. These two fatty acids, typically at least one of them will have a double carbon bond in it, so it'll be an unsaturated hydrocarbon. And then instead of three fatty acid chains hanging down, there's a phosphate and modified amino acid head sticking up that is hydrophilic. In other words, a very polar molecule that interacts with the aqueous solution outside of the cell or the aqueous solution inside of the cell. On top of the phospholipid bilayer itself, in the case of animal cells, we'll have cholesterol molecules. You will not find these in plants or bacterial cells. So this is a diagnostic feature for animal cells. Cholesterol is an animal lipid molecule. You will find in all living membranes of all cells everywhere transmembrane proteins, peripheral proteins, integral proteins, which an integral protein just means it reaches all the way across the membrane. When it's got a pore in it, it's going to be some sort of channel or transport protein right here. Peripheral membranes are meant to anchor some structure to a portion of the membrane, typically chunks of the cytoskeleton right here, and also act as bases for other nearby proteins that may be involved in signal transduction across the membrane. So there's a lot of stuff in the membrane. You've also got molecules either attached to proteins or attached directly to phospholipids that stick out into the extracellular environment and are used for cell-to-cell identification to distinguish related cells that this cell should interact with, to identify foreign cells that the cell should repel or not interact with. Recall that the phospholipids that are the primary component of every phospholipid bilayer, which is the more detailed way of saying cell membrane or plasma membrane, those phospholipids always consist of a central glycerol molecule, just like a triglyceride, and this is a modified triglyceride, two fatty acids hanging off from two of the carbons, one of them again usually unsaturated, sometimes both, sometimes multiple. It just depends. But instead of a third fatty acid hanging off of the third glycerol carbon, we have a phosphate group right here, and R represents some modified amino acid sticking off here. And it is above the molecule right here because it's naturally excluded from this hydrophilic, this hydrophobic region right here. This very, very charged head right here is free to rotate because this covalent bond rotates, and so it'll be up here facing the aqueous solution. All right. Phospholipids do this naturally. They arrange into this sort of bilayer here automatically. If you put them in slightly soapy water with some salt in there, and not soapy water, but slightly salty water with a pH of about 7.3 or so, and shake up a mixture of phospholipids and that salt water, you'll get droplets that look like this, where the phospholipids have arranged themselves in a sphere with all of their tails facing inwards, and this is this water-excluding hydrophobic inner area here. These spheres can only get so large, so if enough additional phospholipids are induced to join this structure, it'll reorganize into a bilayer like this with a water-filled central core, a water-excluding intralaminar, lamella is Latin for leaf, so intralamellar area right here, lamellar area, and then more hydrophilic heads facing the outside, facing the aqueous solution outside. And of course, if that gets big enough, then it can almost look flat. It's a very, very large circle where any small piece of it, you can barely tell the radius. Anyway, a little bit about the nature of phospholipids and how they naturally form these structures. So secondly, as I said before, integral proteins, transproteins, peripheral proteins are all terribly important components of a cell membrane. And they usually have two general big classes of function. One is anchor points for other molecules that are doing some sort of job, and secondly is as means for information or matter, atoms, molecules, and things like that, to move from one side of the cell membrane to the other. So integral protein simply means a protein that is a component of the cell membrane and crosses from one side to the other of it. And so there's a terrific amount of integral membranes, and they have lots and lots of different shapes and functions. 1 and 2 here typically represent some sort of proteins that are involved in signal transduction. Something interacts or binds to a surface of this part of the molecule right here, and that induces a change in behavior across the entire protein. So this part changes, and then that induces effect inside the cell. That could very well be the case for this molecule right here. When you see this very, very large ring-shaped structure right here, this is usually indicative of some sort of pore or channel or transport protein, something that regulates the movement of things across from one side of the membrane to the other. How do I know? It's just because it's really big and it has lots and lots of these alpha helixes in here, making what's called a rosette, of lots and lots of proteins joined together. So this one's made out of several different proteins joined together. In all of these cases, the alpha helixes in here are hydrophobic, and so they tend to help keep the entire protein structure within the membrane. And then here are these areas that have a very distinct third level of organization, non-repeating, non-cyclical. Those are typically going to be a bit more hydrophilic, more interactive with other charged molecules, and that kind of supports their role as interacting with other molecules and inducing a change across the membrane. Recall that your amino acids are divided up into different chemical groups based on the chemistry of their side chains only. Any individual amino acid is automatically an acid and a base at the same time. But when they're chained together, those effects are eliminated pretty much. So then it's really just the chemistry of the side chains that has an effect, because this acid group has been neutralized by being bound to the amine group on the next amino acid in line. And same thing for the reactivity of this amine. It's been pretty much knocked out by joining onto the next amino acid. But the side chain's chemistry is retained. Basic, acidic, aromatic, nonpolar, regular nonpolar, and charged, right? Polar with charged, with partial charges. Carbohydrates also play a role in cell membranes. So far we've got lipids, we've got amino acids in the form of proteins, and then we've even got carbohydrates right here, glycoprotein. So this, is a sugar attached to a protein, a carbohydrate attached to a protein. Remember, the word sugar normally refers to something that actually tastes sweet. That is a subset of the larger group of things called carbohydrates, carbs. Here's a glycolipid, where a carbohydrate polymer is attached to a phospholipid instead. That's the, that's an alternative R group we could have attached right here, a phospholipid, a sugar chain. Where is it? Yeah, on this R group right here. So there's a lot of possibilities as to what that could be, including these glycolipids right here. All right, so a whole lot of proteins on the surface of a cell are what are referred to as receptor proteins. So these tend to transduce information across from one side of the cell, and in some cases, like in the case of an HIV viral particle right here, or these days a COVID-19, a coronavirus in-19, the novel coronavirus virus, which causes COVID-19, which is a disease. Anyway, those viruses have to interact with a receptor protein, which mistakenly, well, on purpose from the point of view of the virus, but by mistake from the perspective of the cell, induces the cell to invaginate in this location receptor-mediated endocytosis, and that's how a lot of viruses get into our cells. So these are CD4 receptor proteins that are part of our identification of self. Membrane fluidity. The membrane needs to be flexible. It needs to be somewhat permeable so that water molecules and gas molecules can get through it without too much resistance. All right, but it also has to be tough enough that it can kind of hold a cell together and maintain the identity of the thing called a cell. In other words, not rupture easily. And that's done in two ways. Temperature is the effect. It tends to, cold temperatures make the membrane more rigid, high temperatures make the membrane more rigid. And there are two ways of modifying this process. In all living cells, you can alter the phospholipid. So saturated fatty acids tend to pack more closely, they get more solid. Unsaturated, unsaturated fatty acids have more space. They tend to be more liquidy at room temperature, and living organisms can alter the fatty acids on the phospholipids to make them more fatty or more oily, if you will, more dense and packed or more liquidy and fluid as a response to temperature. So if it gets really, really cold, they'll tend to remove the unsaturated fatty acids and tend to replace them more with saturated fatty acids to make the membrane more firm, and that is a response to cold temperatures. In animals, we can also add or remove cholesterol molecules. The more cholesterol in the cell membrane, the more the membrane remains it keeps it from getting too fluid when it's hot. So cholesterol is a stabilizer. Adding more cholesterol reduces the rate at which phospholipids switch back and forth from one location to another. Less cholesterol and the membrane is more fluid. So there are two different approaches to changing the lipid composition of the membrane that can change permeability. And in all cases, cells are trying to hold to a steady level. They adapt to keep the same permeability. They don't take it up and take it down for different reasons. All right, so here's an illustration or a movie illustrating membrane fluidity.
Living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
So There is also a polarity to all cell membranes, as this statement right here says they're asymmetric, and that just means that, you know, the the inside has different proteins attached to it than the outside, and the outside is typically where you'll find those glycolipids and those glycoproteins attached. On the inside, we're not going to have the signal molecules because this is inside the cell. It doesn't need to communicate with itself in the same way. And, you know, the proteins interacting with the outside world have a polarity, and that's really all that means. Additionally, we have these exterior proteins, so they're not really illustrating one properly right here. But a exterior protein like this one right here can be attaching to the extracellular matrix, which is going to be collagen fibers and various other fibers of that nature, extracellular proteins. These fibers down here are part of the intracellular cytoskeleton, so these will be actin filaments, as it says right here, cytoskeleton actin filaments. Much larger would be microtubules, and of course intermediate to them would be the intermediate filaments. But, you know, filaments of the extracellular matrix will be on this side, and typically a peripheral protein will be an anchor point for some of that material. And indeed, it's proteins, integral proteins that synthesize those extracellular proteins, because they have to be made inside the cell and then pumped out. So, a huge job of the membrane, and the sort of stuff that's regulated by all membranes of living cells, is the transport of material across that membrane. It is selectively permeable, meaning that it regulates, it differentially allows certain things through and other things not, and it can change that. It can be selective about the process. All right. So, again, you know, the fluid of the cytoplasm, the cytosol inside cells, is inevitably chemically different from the extracellular fluid unless it's one cell inside of another very, very similar cell. All right. So, for instance, living cells tend to have very large amounts of sodium outside of them and low intracellular concentrations, but on the other hand, a relatively high intracellular concentration of potassium. And that is maintained through not just selective permeability, but active transport of materials from one side of the membrane to the other. So that's not in balance. It is an imbalanced situation, and what it does is creates a potential. Anytime, take this just as a rule from here on out, anytime there's more of something on one side of a cell membrane than the other, that immediately allows for the potential of work, because things want to move from where they're highly concentrated to where there's less of them. The physics favors movement in that direction. And if you can harness that difference and turn it on and turn it off, you can get meaningful things done. And all living things do that. So one easy way to tell that a cell is alive is to note that the chemical environment internally is different than the chemical environment outside because of their ability to exclude and move things around. And that movement of things across the membrane is either passive, we allow things to move down their concentration gradient from high down to low, that never requires energy. It may take a tiny amount of energy to open or close a gate, to open or close a pathway, but the movement itself is passive. We're just allowing stuff to go down its concentration gradient. So for instance, allowing sodium to suddenly rush into a cell until it equalizes the concentration gradient across the cell membrane is the first step in a neuron's signal processing, a neuron sending a signal from one area to another. It's the first step in a muscle cell starting to contract. That movement of sodium from high to low concentration, we open up passive channels. Active, on the other hand, is how we set up the sodium gradient. The reason there's a lot of sodium outside a cell and a whole lot of potassium inside cells is that we expend energy in the form of ATP to actively pump things to those positions. So all living cells constantly spew out sodium. They've got all kinds of things that will let it back into them, but they always spend energy pumping sodium out, and they always spend energy pumping potassium in using ATP. That is active transport. Active transport simply means you're using energy. All right, so passive transport and active transport naturally have a couple of different specialized versions that we need to talk about. all passive transport's based on the idea of diffusion. So things diffuse from an area of higher concentration to an area of lower concentration. Lots over here, none here. Still a whole bunch over here, just a little bit over here. So the net flow of material is still from up to down across the membrane. And by, convention, this is always outside the cell. This is always inside the cell. And then eventually we can get to a diffusion will stop, or net diffusion will stop when we reach a iso point where there's the same concentration outside and inside. It doesn't mean molecules aren't still crossing from one side to the other, but the frequency of movements is statistically the same in both directions. 50-50 chance of going in or out. Here, 100% chance of going in. Here, 85% chance of going in, 15% chance of going out. Whatever you want. Something like that. Okay? Very, very small and non-polar molecules are able to diffuse straight across the cell membrane, all right? So the two gases that we spend the most time paying attention to, molecular oxygen, O2, and carbon dioxide, CO2, both easily move across the cell membrane because they're small and relatively uncharged. So they don't interact with the the charged heads. Why? Because they're uncharged. And they don't spend time in here. There's nothing keeping them here. Especially if they accumulate right here, they're free to go ahead and pop out and enter this charged medium right here. They're not going to interact with anything in here. They won't be allowed to really accumulate together in very large masses. They'll tend to be excluded towards the intramembrane space here, but they're allowed to move across without resistance. Also, lipid-based hormones can move straight across the cell membrane without a lot of resistance, and those can even be relatively large molecules. All right. So, what are the things that affect diffusion rates, the movement passively down your concentration gradient? Well, the concentration itself. Oops, there we go. The higher the concentration difference, the greater the rate of diffusion. The smaller the molecules, the greater the rate of diffusion. This is an easy one. Put a bowling ball on the ground and a tennis ball right next to it. Kick both of them with your foot at the same intensity and see which ball moves further. Temperature. Molecules move faster when the temperature is higher, so diffusion occurs faster at high temperatures. It happens lower at slow temperatures. Solvent density. If we dehydrate the cytoplasm, that can change diffusion rates generally because, if we lose water, everything dissolved in the water is becoming more concentrated. Solubility. Non-polar lipid-based molecules will diffuse faster through liquid because they don't interact with the water molecules. They just slide down their concentration gradient. If they are polar, highly dissolvable molecules that, interact by dissolving into water through charges, excuse me, they tend to get slowed down a little bit because they're sticking and being held by one water molecule, which is also partially being held in place due to interactive forces amongst water molecules, and so movements slow down a bit. The greater the distance, the slower the diffusion rates. This is why one of the reasons cells can't be over a certain size. If they get too big, the diffusion distances become so great that the rates become slow, and that in turn inhibits the cell's ability to function. If we add pressure, we can change the movement of materials across membranes, including reverse osmosis, which if you apply enough pressure to liquid, you can send salty liquid across a semi-permeable membrane that's permeable primarily to water, and only water will come out the other side. The salt will be held behind. That's reverse osmosis. And so the point is, some tissues in your body, particularly in your kidneys, do use hydrostatic pressure, and your circulatory system does this as well, to change the movement of things across membranes. All right. Now, so what do we do to allow things to move passively that are too big to pass through the cell membrane? So things that are larger than a bit of water, your gases and lipids, or more charged than those things. Water tends to be more charged. We've either got channel proteins or carrier proteins, and these are simply proteins that allow for a bigger hole punching through the cell membrane to allow a molecularly larger structure to go through. That's what both of them are for. They still allow things to diffuse. They're not actively moving anything. They're just allowing stuff to move that otherwise couldn't cross the membrane because the molecules are too large the ones we're talking about diffusing, or they're too charged and would stick to the heads on either side right here. For those molecules, we're going to either have channel or carrier proteins. The difference here is a channel protein just opens up and allows anything as small as it or smaller to slide right on through. So it's not as selective as a carrier protein, which usually has a very, very one-to-one relationship or a two-to-two relationship with with a couple of different molecules, and only very specific molecules can interact with this protein, and it literally changes shape. It kind of rolls in, rolls out to move things across the cell membrane down their concentration gradient, and that's a carrier protein. It is much more selective. Much more selective. Think of like a super complicated, soft, floppy, revolving door, and that only certain things could fit into the flops of the revolving door. Whereas a channel protein is very much just like opening a door. Open the door, anything that fits through the door goes through, and then when you close the door, it's closed. All right. So the channel proteins, all of these are going to be integral membranes, which means they just go all the way through the cell membrane. Integral membrane, integral membrane. Integral just means all the way through the cell membrane. It doesn't tell you anything about its shape, structure, or function. That's a different question. All right. So these integral proteins will inevitably have to have these hydrophobic regions that are part of a core. That inner core is hydrophobic. This should say hydrophobic amino acids, and then the top and the bottom, the bits facing the outside and the inside, are going to be very hydrophilic, and they're going to attract ions and other polar molecules, right? That's going to be the nature of these channel proteins. The central portion going right through the cell membrane is made out of, it typically is going to look something like, where was the picture? Like this. And this is a ring of alpha helixes. The pale blue is the back side of the ring, the dark blue is the front side of the ring right here, and implicitly there's a hole going through the middle right here, and this thing can open and close and interact with things in some way. And that's what we're talking about when we talk about a channel protein. All right, and so here's an example, an aquaporin. It's specific to H2O in that there's the only things smaller than water itself are going to be, well, oxygen. An O2 molecule is bigger than a water molecule, so O2 won't go through, but hydrogen ions themselves can pass through an aquaporin, so they will flow through. But otherwise, H2O is about the smallest thing that you're going to encounter trying to move through. There's one or two ions that might challenge it. And so we have an awful lot of these aquaporins, and so that allows water to move more rapidly or less rapidly across the cell membrane via osmosis, the special case of diffusion of water. Osmosis is made easier if we have lots of aquaporins. It's made slower and more difficult if we have fewer aquaporins. Why? Because water molecules themselves are very polar. They tend to stick to things. That's why we need a pore. Now, a carrier protein, on the other hand, as this illustration shows, changes shape in a very, very specific and choreographed manner to allow a particular molecule, but no other molecules, no other substrates to pass through the channel protein. So here we've got a passive glucose transport protein that allows glucose to go down its concentration gradient from outside the cell to inside the cell right here. Right, and this is passive. It doesn't take any work, but it won't let other molecules through, just glucose in this particular case. All right. We already know that osmosis is a special case of movement of water across the cell membrane, always from the point of view of water higher concentration, less salt, less sugar, less everything else, to an area of lower water concentration, area where there's a lot of salt, a lot of sugar. Another way to think of this is wherever the most sugar or salt is, or anything else that dissolves in water, so proteins count as well, water will move towards that stuff, right? Water follows salt, water follows sugar, water follows proteins, right? If you've ever been to the beach at the sea and had a wet, salty towel, did the experiment where you tried to dry your saltwater soaked towels, they dry really, really slowly in the same air that a freshwater washed towel will dry really quickly, and that's because the salt is pulling water towards the towel from the air around it. It's resisting the loss of water. Water follows salt, and that's the basis for osmosis. When we have that movement of water across a membrane that blocks the salt, That's specifically the situation of osmosis, and that's how all of our cells gather water. All right. So for that to happen, our cells need to be hypertonic to the environment around them, or the environment needs to be hypotonic, less salty than a cell. All right, so a cell needs to be hypertonic to its environment, or another way of saying the exact same thing is that a cell needs to be in a hypotonic environment. The world it's in should be hypotonic to the cell. That means water will flow towards the cell via osmosis. The cell itself must be hypertonic to its environment so that it draws water towards itself. When it's in balance, that's called isotonic, which a lot of cells work isotonically. Many aquatic marine creatures in the oceans are pretty darn close to isotonic with their environment. Usually they're just a tad more hypertonic than their environment, so that ensures their cells always remain fully hydrated. They draw all the water in they can. All right. Why is this important? Well, if you dehydrate cells, you lower the water concentration in there, you're really changing the chemistry that proteins in there are exposed to, and those proteins can change shape to a point where they can't really function anymore. Not to mention the fact that in the case of blood right here, if you remove lots of the water from the plasma and lots of water from the cytoplasm inside the red blood cells, you get a thick, crispy, chunky sort of mix of stuff that can't flow through capillaries anymore. It'll all get clogged. On the other end of the spectrum, we can't allow our blood to become too hypotonic to our red blood cells. If that happens, so much water will flow into our red blood cells that, and them being animal cells without a cell wall around them, they'll burst. Remember, this problem tends not to happen in plants. They're very resistant to this. Algae, bacteria, fungi, all of those cells are generally very resistant to this phenomenon. They can, exist in a hypotonic environment. They'll suck up all the water they need, but they can never overinflate because of the cell wall. That will keep them intact. But all cells are capable of being desiccated to the point, and they're, by extension fluids, desiccated also to the point where they really don't function properly. So animal cells tend to function best when extracellular fluids are isotonic. The fluids immediately around them, there tends to be in best health really a gradient from very hypotonic outside the animal. That's why we drink fresh water, not salt water. Much more hypertonic inside the body tissues, the blood, but then the cells and tissues themselves are just a little bit more hypertonic than the blood itself, so water tends to be drawn out of blood and into animal cells all over the bottom until they get to kind of a balance with the blood, and that's good health right there. So just to explain that term a tad more. All right, so here we're looking at plant cells, and again, plant cells are quite happy when they're in a hypotonic environment, when the liquid around them is hypotonic to the cells themselves, because they can just inflate until they're puffed up tight like a balloon against their very, very tough cell wall. There's no tonic situation that I'm aware of that can create such a pressure inside here that the cell could rupture its cell wall. The cell wall's too strong for that. So that's not really a problem for plants. However, if they become dehydrated, if they become desiccated, and material water fluid is drawn out of the cells, what happens is the cell membrane pulls away from its connections to other cells. Plants rely on cross connections between one cell and another, so they'll rip and break those connections, and that's usually a fatal event. So if you desiccate a plant, and for the same reasons an animal cell, they're going to die too. If they become too concentrated, too little water, too much everything else dissolved inside right here, they're going to malfunction. They're going to rip themselves to pieces. And there's a point where you can't come back and you can kill the cells here. All right. There's an image right here, but it's not showing up. We won't worry about it. Okay. Cellular osmoregulation. So this is a paramecium. It's a freshwater, animal-like, single-celled eukaryotic organism. Eukaryotic, it's got a big nucleus inside. It's got mitochondria. It's got all kinds of organelles in here. And in fact, it has these neat things called contractile vacuoles right here. This cell is naturally salty with sugars and other stuff like that inside it. And because it's an animal-like cell, it's not surrounded by a cell wall. So it would overinflate, swimming around in a freshwater pond. But that doesn't happen because it has these specialized vacuoles right here, which are surrounded by basically the fibers, the protein basis of muscles. So at a subcellular level, they're muscularized vesicles, if you will. It's not made out of cells. A muscle is an organ made out of cells. But these are the components, the molecular components of muscles surrounding these vacuoles, and it causes them to squeeze periodically, and they've got a one-way valve that leads out of the cell. And so these things suck up excess liquid from the cytoplasm of the cell and pump that liquid out using regular sequential molecular contractions. So this thing this is a single cell that's got its own kidneys, if you will, that do the same job your kidneys do of extracting excess liquid and pumping it back out of the cell to keep it from overinflating and bursting. That's really neat. That is a really, really neat mechanism. And you can watch these under a microscope. It's so fascinating. Put them in salty water and the contractiles, put the animal in fresh distilled water and the contractile vacuoles pump like crazy. Put the animal in somewhat salty water and you can find the salinity of the animal cells by adding salt until the vacuoles stop pumping, at which point the animal is isotonic with its environment. It's such a neat laboratory experiment. One day, hopefully you'll get to do that somewhere. Not for us this year because we're online. Marine animals tend to be, somewhat in matching their environment, as it said right here. But freshwater animals inevitably have to somehow get rid of lots and lots of water. This paramecium has to do it at a cellular level. This fish does it, by getting rid of lots of urine through the function of its kidneys. We maintain our blood chemistry to regulate water levels in our blood, and we have osmoreceptors on cell membranes of brain cells that regulate that stuff and send off hormones that change the way our kidneys function to make sure that we get all the water out that we need to, or keep the water in that we need to. So, that was an awful lot about passive transport. So remember, there's two ways: simple diffusion straight across membranes, or for large molecules or charged molecules, we'll have to have channel proteins or transport proteins to allow things to diffuse across the cell membrane. Now, the way to concentrate things on one side or the other of the membrane is by using active transport, and we can pump stuff out of a cell to increase its concentration outside or decrease the internal, or vice versa. So we have pumps pumping things in both directions. It totally depends on the cell, the organism, and the situation. All right. One thing that's almost continuously done by all living cells is they tend to pump hydrogen ions out into the extracellular environment. All right, they always push them out. And a couple of other things that are constantly done: all living cells exclude sodium. They pump sodium out of the cell, they pull potassium in. That's, again, a good way to tell that a cell is alive, if you can measure those sorts of things. All right. And the pumps are always proteins, and if we have energy, if we have a pump, the word pump, requires energy. Every pump that you've ever tried to use is either hand-powered, it requires muscle power, or it's electrically powered, or it's engine-powered. A gasoline engine drives the pump, but you know that a pump requires energy. So in you and every other living thing, that's typically ATP. In a lot of cases, what we'll do is actually use ATP to pump something else like hydrogen, and then hydrogen moving down its concentration gradient. That's the secondary type of active transport right here. The hydrogen ions power the movement of something else. That sounds complicated. It is. It's a slightly complicated subject, but you're going to have to be able to talk about it. So let's make sure that we get active transport, particularly the more complex version, using a secondary power source. Let's get that worked out. So when talking about secondary transport, it's really important to get an idea of what we mean by an electrochemical gradient. So a gradient is simply a difference of concentration of something from one side, from one point to another. And electro means electric, like charge, and chemical means the actual number of atoms of something. So here we're looking at a situation where we've got a cell membrane, and this is the extracellular fluid outside of the cell. This is the intracellular fluid inside the cell here. And as I told you before, all living cells normally keep most sodium high in extracellular fluid. The concentration of sodium is quite high. In you and I, it's about 120 milliosms. And the potassium concentration is a similarly large number. It's a bit less inside the cells, though. So potassium is pumped in and sodium is pumped out. And that creates a chemical gradient. There's a sodium gradient from outside to inside. There's a potassium gradient from inside to outside. Chloride ions, which come with salt, you know that you eat sodium chloride. Chloride ions are generally excluded from transport across the cell membrane, being negatively charged, that's possible. And so we have a high concentration of sodium and chloride outside, but a very small amount of chloride internally right here. So we have a net positive charge on the outside of the cell and a net negative charge on the inside of the cell right here. Sorry, and that's due to the large proteins. I got ahead of myself right here. Not much chloride, but lots and lots of negatively charged proteins, huge numbers of them. And you'll recognize that there aren't a lot of proteins out in the world in general, floating around in the ocean. They're all kept inside cells. If they're found out here, they're immediately taken in by living cells as food. So the proteins are kept and the amino acids are kept in high concentration inside, and they're generally negative in charge. So there is a net positive charge on the outside of most cells and a net negative charge on the inside of a living cell. So every cell acts kind of like a battery membrane as well, and there's the potential for current to flow across the cell membrane through the movement of charged particles, and that is an electrical chemical gradient. So a little bit about how we move things across membranes, some other stuff. We've got these integral proteins. Remember, an integral protein simply means a protein that crosses the cell membrane. It's got to have positively charged regions here, hydrophobic regions in the middle, and charged polar regions on the other side here. And that's how it holds itself in the membrane properly here. All right. And we can classify these carrier proteins based on how they move stuff across the cell membrane. So a uniporter just moves one thing in one direction. A symporter, sim for the same, that's why symphony, everybody's following the same sort of rules. Both things move across, two or more things, but it's typically two, move across in the same direction. That can be both in or both out, it just totally depends. And then we finally have antiport transport, where one thing is moved in and the other thing is moved in the other direction, whatever the particular details are. So these are three general sort of categories right here: uniporter in or out, symporter in or out, antiporter in or out. A may go in, A may go out. I can think of examples in both cases. All right. So now going back to this idea of transport either being primary or secondary. Remember, primary transporters use ATP directly to power movement. Secondary uses something that was originally transported using ATP, but it uses the power of something else that was previously transported as the power source for its transport. So let's go ahead and take a look at this. Active transport. We're looking at a really important feature of you and I and every other living cell out there, and that is the sodium-potassium co-transport protein, and it is a case of an antiporter. The sodium will continuously be pumped out of the cell, and the potassium will continuously be pumped into the cell. And that is the sodium-potassium pump. And it inevitably moves 3 sodium out for 2 potassium coming in. And notice more positive out than positive in, so that alone tends to favor a negative charge inside the cell. Here's that negative, and a positive net charge outside of the cell, because we're always pushing out more positive than we're pulling in. And moving those 5 atoms right there requires 1 ATP. So the question, is this pump uniport, symport, or antiport? Sodium out, potassium in. And it's an electrogenic pump. Genic means to generate, and electric means, charge. So here's a case of secondary active transport, and this is a big, big deal in you, all right? You pump sodium out of most of the cells in your body, and really importantly, pump them out of the cells lining your digestive tract, all right? So you pump these sodium ions out, so they're in a high concentration outside here. So if you open up a gate that will allow sodium to attach, sodium's going to want to come down its concentration gradient back into the cell. It's also drawn in because sodium is positively charged and there's a negative charge inside the cells. So it both is pushed by concentration and pulled by electrical gradient. So that's two reasons for sodium to really move into a cell aggressively and quickly. And this is a co-transporter right here that binds to a glucose molecule, and that's how you get glucose absorbed in your digestive system. You make salt water. If you drink perfectly fresh water, starting with your stomach, but certainly your intestines will pump lots of sodium into that water and create a salty water, and then that sodium can be allowed to move back down its concentration gradient and take monomers of various foods you've digested and bring those in at the same time through those co-transporters. Some of the best illustrated and best understood ones are glucose-sodium co-transporters. And since sodium and glucose are both moving in the same direction, it must be a case of a symport transport right here. And the power source is not directly ATP, but the reason the sodium is all piled up out here is because of ATP that the sodium-potassium co-transporters are using. All right. Now also, there are mechanisms for moving much larger things in and out of cells. Animal cells in particular are capable of doing this. Plant cells, fungal cells, and a lot of protists can't. But that is bulk transport, where we can move very large materials across the cell membrane. That requires that the membrane be more directly exposed to the outside environment. So this is a characteristic that animal cells are specialized at, predatory cells. All right. And so there are three forms of bulk transport, as we say right here. Phagocytosis, which is the attaching to a single large object and invaginating that object, to swallow it, if you will. Penocytosis, the regular importation of vesicles full of liquid from the outside world with whatever happens to be dissolved in that liquid. And, so think of it as, a cell taking gulps of the water around it, if you will. Whereas phagocytosis is a cell swallowing a sandwich whole. And typically a white blood cell swallowing a bacterial cell, or the free living version of that same thing, an amoeba swallowing a bacterial cell in a pond somewhere. Same deal. And then we have receptor mediated endocytosis. That's when we have receptors on the surface of the cell, and when enough of the substrate that they are specialized to attach to attaches to them, that causes the cell to initiate an invagination. Sort of like penocytosis, except it's based on having attached to a required number of the thing that we're trying to swallow, and only at that point will the cell swallow a gulp of water with all of those molecules attached to the receptors. And that's exactly what I was alluding to back here in the presentation when we're looking at this virus sitting on the surface of a receptor on a cell. We'll get to it eventually here. There we go. So this was the HIV virus right here. It's binding to, this is not here to detect HIVs and cause them to come into cells. What's happening is the virus is tricking this molecule and using it to fool the cell into invaginating and pulling the HIV virus in. But that's kind of the way receptor-mediated endocytosis works. The target binds to a receptor on the surface of the cell. When enough of these are stimulated, the cell invaginates and pulls in as a vesicle a drop of liquid with all of that stuff in it. Receptor-mediated endocytosis. All right, so here's some illustrations of these processes at work. I've got a couple of pictures that just aren't working. Hold on a second. So this is from Wikipedia. Let's play this animation of a neutrophil, a particular type of white blood cell. I'm sorry, it's in Cyrillic right here. But originally it was in English. A white blood cell, a neutrophil, phagocytizing bacterial cells, not these larger things, these are red blood cells right here, but the bacterial cells, smaller things around here. Let's see what happens. So a whole bunch of your white blood cells work in ways that if we found it out in the wild, we would say, oh, that's kind of like an amoeba. That's exactly the idea. So these are, your immune cells that are specialists at absorbing bacteria. And so here we see it's run over one or two over here and swallowed them. it's moving around searching for whatever it's interested in. It looks like this may be its target. In this particular case, let's see. And what they do is they swallow anything foreign they find, internalize it, and if possible, they'll digest it using the hydrolytic enzymes in their lysozymes, or they'll deactivate it using the oxidizing enzymes in their peroxisomes, or if nothing else, they'll just hang on to that subject and surround it for however long the cell lives, taking it out of circulation. So this phagocytized that object, whatever it was, and it's being digested. So that's an illustration of the process of phagocytosis. Pinocytosis, again, just drinking drops of liquid here, and with whatever happens to be concentrated in that liquid, some of it statistically coming in with a vesicle full of that liquid. And, that's a way for cells to bring in samples of the outside environment without specializing what it is they're bringing in. And then finally, receptor-mediated endocytosis. We have to have these receptors. The target binds to them. When enough receptors have been triggered, the endocytotic event occurs. It involves contractile proteins like you find in your muscle cells and cytoskeletal filaments from your cell that that regulate this process of making a vesicle. Fascinating subject all on its own. But anyway, and then you have a vesicle inside the cell with a large cargo of whatever it is we were after. A lot of cells recycle the neurotransmitters they release this way. So they're released into an environment, they bind to their targets, and then the excess is swept up by the cell that released it, pulling it back inside where it can recycle the neurotransmitters that that it released. That happens quite a lot in you every day, all over the place in your physiology. And then also we have exocytosis. So a great example is this is how you release neurotransmitters from a neuron to a muscle cell or a neuron to the next neuron in line, or how you release digestive enzymes into the fluid, extracellular fluid that is the fluid inside your digestive tract, in your intestines, in your stomach. It is by exocytosis of these vesicles full of hydrolytic enzymes in this particular case. So you do a huge amount of work with exocytosis all day long, every day, in order to stay alive. Not every cell in your body does this, but a whole lot do that maintain critical functions that are required for you to continue functioning. So the bulk means of moving things across cells. Endocytosis, the movement of things in. Via phagocytosis is when you're, for very large subjects. Pinocytosis, just taking samples of the liquid. And then receptor-mediated endocytosis, where we take in targeted molecules. So 3 ways of pulling things in. And as a generalization for y'all right now, 1 way of moving things out, and that's via, exocytosis. And that's the end of the presentation on chapter 5 in your OpenStax textbook, the chapter on membrane structure and membrane function.